I will start talking about why we are here today. So thanks a lot. Cecilia and myself are a member of the sedaDNA Society, and we spent lots of time over the last months to create this initiative and create this working group. So here today we are here together to have our first symposium, and I will describe very, very quickly what we want to do with that to let the place to speaker to do the talk today. So we call this working group African sedaDNA working group. And I will first describe what is sedaDNA, because maybe some of you don't know yet about it. So sedaDNA stands for sedimentary unchained DNA. So this is basically a DNA molecule you can find in environmental archive. So you can see on this slide here a lake, an ice-covered lake, where scientists go to sample sediment. So sediment is what you have archived at the bottom of the lake. And you can retrieve many different proxies from this sediment, including DNA molecules that are preserved over time in the sediment, and you can use them to authentify different types of organisms. And Beth will talk a bit more about that just after our introduction. And because many scientists in the world use this tool now, we created earlier this year the sedaDNA Scientific Society. We are no more than 230 members, and we are really satisfied about that because many experts belong to this society. And the first initiative we started with this society is to create this working group. The reason why we did that is because in African lakes, you can retrieve a DNA in sediment also, and many good researchers from our society already published paper about that. And most of them are here today to present their work. When we created our society, we realized that in the map, we didn't have any member from Africa. And what we would like to do with this work is to empower African researchers to lead research in this specific area. So we now have the skills and the expertise to support African researchers to do such work. The challenge and limitation of using sedaDNA in lake with one temperature, but it's something we will talk about later. So yeah, we are here for that today, to give African researchers a central role in African lake sedaDNA research. And it will be Cecilia time now.
So hello everyone, and thanks again for joining us. So I'm not gonna go over the program, but I'm just gonna explain you really quickly why we organize this symposium. So really the goal of this symposium is to set up the ground and use it as a foundation for to create the African sedaDNA working group. And today's seminar will hopefully provide an overview of the sedaDNA research and what kind of information and research question it can answer, as well as provide an overview of the current state of sedaDNA research in Africa and ongoing research projects that are being led in Africa. So we hope that after this seminar, we'll have enough content to be able to feed the discussions that will be starting tomorrow and during the breakout room session and the brainstorming session of tomorrow's session. Yeah. And so so if you can go on the next slide. So yeah, today we really have really short presentation of 5 minutes presentation, and after each presentation there will be 5 minutes of questions, and because those are very restricted times, to make sure that we're not going over our time schedule, we are going to ask you to use the frame pad. So you just have to click on the link that Eric sent just now. So if you click on it, you will access to a page where everyone can add comments on it, and everyone will see the comments being added on live. And so for each talks, you can write down the questions or thoughts that pops up in your head while the talk is going, and then we can use it as a startup discussion, or to make sure that everyone can have like time to ask their questions. So don't hesitate to use this pad to ask your questions, because obviously we won't be able to have everyone asking the question in live during the 5 minutes after each presentations. Just a quick little point on this frame pad. You have a little, in the right corner, you have a little icon with a number, and there, if you click on it, you can write your name and choose a color, and then we can recognize you as you write down on the pad. But it's not an obligation. You can just do it if you want. So yeah, I think that's everything, and I will let Eric take the lead on the first part.
Great, perfect. So we are already late. We are super good with this kind of thing. We are lucky to have Professor Beth Shapiro today to do introducing about this field of work. So I will let you share your screen, Beth. And Beth is a professor at the University of Santa Cruz in California. Yeah, the opposite, the University of California in Santa Cruz.
I can share, but you have to stop sharing first.
Okay, sorry.
Thanks. All right. Now let's hope this works. Keynote. All right, let's see if this will, or maybe I have to. How do I play now? Oh, here we go, play. Do you see this? Yes?
It's perfect.
Excellent. So I have the daunting task now of introducing this whole field in 10 minutes, which is, I'm honored to have been asked to do this, but also a little bit scared, and I will try not to run too badly over time here. I'm really just going to introduce some ideas and concepts that I hope will be discussed more thoroughly as we get into the breakout rooms. And I know that you're going to hear a lot about these different things as we move forward. So environmental DNA, as we know, starts with a sample in the background, and then you have your sample, you collect it, you extract DNA, and you do something to generate the data. You might do metabarcoding, you might do enrichment, you might prepare that extract into a sequencing library and do shotgun sequencing. And we can do all of these things, and you'll hear more about these as these seminars progress today and tomorrow. And then you get data and you come up with some way of asking interesting scientific questions about the data that are there. The field is young, there is a lot still to learn, and there are a lot of challenges, and just some of them that people are beginning to explore. And I'm not going to go through this at all. This is just what the field is thinking about. How do any of these choices that we make impact the diversity of data that we're getting from these samples, the enzymes that we use, the primers that we choose, how deeply we sequence, exactly how we enrich. These will all affect what data we can generate. And so we need to think hard about the decisions that we're making, both in terms of collecting samples and in generating data when we're designing our scientific questions. And of course, we're here not just talking about environmental DNA, but ancient environmental DNA or sedimentary ancient DNA, called both of these things. This is just a picture from one of my field sites in Alaska, where what we see is a sedimentary layer. There's a lot of ice and frozen dirt there, and there's a chunk of sticks and wood and leaves and stuff that I've pointed a yellow arrow to. And that we know is around 10,000 years old. And we think this is the time period when people first became really abundant in the region. And we might want to ask, for example, what is the impact on the diversity of plants and animals? And to answer that question, we would extract DNA from below that layer and above that layer, and then compare what plants and animals are present. And this is just a touch, a sense of some of the questions that we might be able to ask. Of course, with ancient DNA, we have additional challenges, scientific, technical challenges. The sequences of ancient DNA are short. They tend to be damaged. They're harder to amplify. We know that some taxa might be preserved more readily than others, and this might also affect what we can do. And then there's the challenge that perhaps DNA can move up and down the sediment column. So we want to be able to have a robust chronology to know how old the sequences are that we're generating, and also that we don't have movement up and down that might contaminate older samples with younger DNA, as we know can be a problem. So additional challenges associated with this. But why do we care if it's so hard, if there's so many outstanding questions? The reason is because sedimentary DNA and ancient DNA present these amazing opportunities for discovery that we don't have without these technologies. The past, by its very nature, is different than the present day. And so if we come up with these ways of being able to look directly at the past and ask what's going on, we are perhaps guaranteed to make new discoveries that we can then use to really enrich our understanding of how the world works, about how climate impacts biodiversity, about how communities are organized, about what makes some communities more stable or resistant or resilient in the face of climate change and anthropogenic impacts. And that is really what we want to know as scientists. So how did this field begin? Well, one of the first papers using ancient sedimentary DNA was in 2003. I was part of this paper. Eske Willerslev did this work when we were at Oxford. And he was also in Copenhagen. And we generated these data. We looked at 5 different cores and we generated sequence data from these cores. And we used an approach called molecular cloning. And probably no one on this call is actually old enough to have done this approach where you have to actually get bacteria to open up and then eat a tiny piece of DNA, and then you sequence those bacteria using Sanger sequencing. And to give you an idea of the size of this data set, here's a table from this paper that shows at the bottom the total number of sequences that we had. 32 sequences for one core, 37 sequences for another. This is absolutely nothing. And yet we came up with this idea that diversity was changing over time and that climate was driving these changes on pretty much no data, that it took forever and was extremely expensive to generate. So this is how it began. And then next generation sequencing saved our life. So we have this way of generating millions or billions of sequence fragments for just part of the cost and almost none of the time that it took us to generate these data sets in the early 2000s. So what kind of questions can we ask with next generation sequencing? And just 4 quick ideas that I think hopefully will bring us up to where we're going to go for the rest of the seminar. We can look at how humans have impacted a landscape. Here was a paper that was published in 2016, where focusing on this part of North America that's called the ice-free corridor that sits in between the Laurentide and Cordilleran ice sheets there. There's a B in there. People were wondering, when did people get there? And did people use this fragment? And using metabarcoding approach, this group led by Michiel Petersen sequenced a whole bunch of DNA and were able to show how both the plants and animal communities changed as that corridor opened up and became ecologically viable. And they were able to show that it wasn't viable until well after people were already established to the south of the ice, which confirmed, in fact, that people used a different way of getting from Alaska down into the rest of North America. They probably went along the coast. Another paper that was recently published used metabarcoding again to look at a hot place, Hall's Cave in Texas, and they had samples that spanned the transition out of the last ice age into the Holocene. And again, looked at, these are these triangle samples on the plot here, spanning from one of the warm periods in red through the Younger Dryas, a cold period, that's the dark yellow, and then the early Holocene, another warm period in light yellow. And we're able to show that we had massive turnover and the disappearance of nearly all of the large mammals that were there. Again, really diving, using ancient DNA into the idea of how climate change was causing not only extinctions, but also the redistribution of diversity in the region. We can also use shotgun sequencing. So instead of using primers and amplifying just some parts of the DNA or specifically targeting something like plant chloroplasts or mammal mitochondria, you can just sequence all the DNA that's there. And we did this a few years ago in St. Paul Island in Alaska, where the second last living populations of mammoths were. We went out onto this lake during the winter and stuck a big coring apparatus down into the dirt. And then we extracted DNA along that dirt and created a robust chronology, an age model. And we're able to show that mammoths survived there until around 5,700 years ago. And at the same time that mammoths disappeared, the lake dried up and became super salty, suggesting that mammoths disappeared from St. Paul because they ran out of fresh water. And then even more recently, we went to an even hotter place, a cave in Mexico, and sequenced a whole bunch of DNA using shotgun sequencing. And here we're actually able to generate enough data to be able to do a genomic analysis of an animal that was there. And this tree here shows now we have broken up little tiny pieces of DNA that we can't use to make a whole genome. And so what we did with each one was we asked, okay, where along this tree is this piece of DNA most likely to go? And sometimes it would go in multiple places, and sometimes it would only go in one place. And we were able to show that this animal whose genome we were sequencing from this dirt in Mexico was a giant short-faced bear, which wasn't even supposed to be in Mexico at this time. So this is just really showing that this approach can lead to some tremendous discoveries. And we're going to talk all about this over the next couple of days. And just one little fodder for thought here. What is going to be really important is approaches to be able to enrich our samples for the DNA that we're interested in. A paper that was published a few years ago was looking for Neanderthal and human DNA in cave sediments, and they sequenced tons of DNA from each of their sediment samples and showed that nearly everything that they recovered was microbial in origin. So they were interested in humans and Neanderthals, needed to come up with a way to do this, and they developed an enrichment approach. This paper actually did lots of things, but this is the one thing I wanted to highlight here, and showed that when they enriched, and here they call it captured, so you see the captured versus not captured, the number of sequences that they were able to generate that were useful to them, it was really, really high compared to without. So only in the tens with no enrichment, but in the tens of thousands after enriching. So this is going to be an approach that we really have to think about developing, especially when we start working in hot climates. So this is all I'm going to say as my intro. Just please remember that this is hard to do. We know it's going to be hard to do, but it's exciting because it really is opportunities to discover new things that we didn't know about before, and to really push science and our understanding of human impacts and climate impacts and what makes communities resilient in the face of all of these things that will help us to develop scientifically based policies for preserving and protecting ecosystems as we move into the future. So thank you very much. And I will stop sharing.
Wow, thank you very much. We can say that an introduction, it was crazy. And I didn't realize you were in all big paper in this field also, all of them. That's very great. Okay, we can open a question for actually 5 minutes, and you can ask any question, and the more naive you can, of course. If you have no clue about what is sedaDNA, ask, and Beth and us, we will try to answer. There is no wrong question there. So please raise your hand. Yes, Alassane.
Thank you, Shapiro, for your presentation. I have a question for you. I would like to have an idea about meta-barcoding and metagenomic approach. What is the differences between both approaches?
That's a really good question because it's super important. And I started off by saying when we decide how we're going to design our experiments, we have to think about what approaches we're going to use. So meta-barcoding means that we're looking for something specific. Like say we want to know what the plant diversity in our sediments is like. People have developed these primer pairs, barcode primer pairs that amplify a region of the plant chloroplast genome that is sufficiently conserved along the tree of life that you can actually put primers in a particular place and variable in the middle. So you can amplify lots of different plant species and then identify exactly what they were based on the sequence in between these two primers. So you'd get a whole bunch of the same locus from lots of plants. And that way you're not going to amplify microbes, you're not going to amplify humans, you're only going to get plants. In metagenomics, you're sequencing everything. So rather than use primers and go in there and amplify just a particular barcode locus, you sequence everything that's in there. So I might get the chloroplast, I might get the rest of the genome, but I'm probably going to get a ton of microbes. So the cost of doing that approach, of doing the sort of amplification of everything, is that most of the data that you recover are not necessarily data that you're interested in if what you want to do is look at the plant diversity or the animal diversity. The benefit of that approach compared to meta-barcoding, especially for ancient DNA, is that in order for a thing to be amplified using this barcoding approach, you have to have a sequence that's as long as those two primers and the thing in the middle. But because ancient DNA degrades, most of your fragments are going to be very small. So in the St. Paul paper, for example, the project, we tried first to amplify mammoth DNA using a metabarcode primer. We were trying to look for mammals. And so we used the regular primer, which amplifies something that's maybe 150 bases long. It's not exactly that, but maybe 150 bases long. And we got nothing. We got absolutely no data whatsoever. But when we just shotgun sequenced everything, metagenomics, and sequenced everything that was there, we got mostly microbial DNA, but a ton of mammoth DNA, but the average fragments were only 50 bases long. So they were way too short to have been amplified using that metabarcoding approach, but they were there, and we were able to get them with metagenomics or shotgun sequencing. And this is why I was saying that enrichment is going to be important. We don't want to spend all the money and just sequence all of the microbes that we can't identify. We want to amplify only the plants or only the mammals. And in order to do that, we have to develop ways to do that that don't involve amplifying long fragments of DNA that are not likely to be preserved when the sediment is hot and wet or from an unstable or variable, temperature variable environment.
Thank you. Thank you very much.
Great. You can ask question in the pad. So I put back the link for people that arrive later. So you can write the question down in the pad and we can try to answer them also later. So we have another question from Rishan that is based on all the approaches you spoke about. Which one is the most cost-effective method for unchained DNA and producing the high-quality result?
That is, again, a really good question. And the answer is, unfortunately, we have no way of knowing until we know something about how well preserved the sample is. What we tend to do is we will do the DNA extraction and we will amplify just a little bit of metabarcoding and then look at the distribution of how long the fragments are. And if the fragments seem to be long, then we will try metabarcoding because that is cheaper if we're interested in doing sort of community ecology things, asking what plants and animals are there. It's less expensive, but it's also, you know, less likely to work with ancient DNA. And so enriching is more expensive. It costs a lot of money to develop the panels that one might use to enrich things. Often this is done by creating short bait molecules. These they're kind of like very specific fishing lures. So I'm interested in getting exactly this piece of DNA from a mammoth. I'm going to design a matching sequence of DNA and add it to my library and hope that it hybridizes to mammoth DNA in there that I can then pull out of the rest of it, wash the rest of the stuff I'm not interested away and only sequence this. So I'm saving money by not sequencing the rest of that gunk, but it's costing me money to generate this bait library. So yeah, it really depends on how well preserved things are and what question you want to answer. But we're lucky that there are lots of different approaches that we can evaluate when we're designing our experiments and figure out what the best way to move forward is.
Thank you, Beth. So we have to go to our next speaker. And again, in the pad, you can put your question and we can reply there if you have more questions for Beth. Thank you very much. It was a very nice introduction. We will continue now with Laura Hepp, Dr. Laura Hepp from Constance University in Germany. If you can share your screen, Laura, that would be great.
I will. Thanks a lot, Eric. Just a second, I'll share my screen. And I will try also to be extremely short. I'm very brief, talking about what I said now, prehistorical biodiversity recorded from sedimentary DNA from Africa and experiences from different sites and preservation conditions. And so the thing is, just a second, the thing is that I started actually working in sedimentary ancient DNA exactly in Africa and African sediments. And so basically the most difficult places that you can start. So I'm going to just talk a little bit. I'm going to give just a tiny hint of the different things that I did, starting with my PhD. And so these are different places that I'll be talking about. So this is for my PhD. And I have to say, Beth, I am actually among those in the room who know what cloning is. And all of this was still based on cloning. So in my PhD, I analyzed sedimentary DNA.
Laura, there is another Laura also that knows what cloning is.
We all do, exactly.
Guys, you're giving away our ages, stop.
No, never, yes, we still sometimes, you know, we know how to clone. Anyway, let's continue to be brief. So this was from my PhD from different lakes in the east and in the Rift Valley in Kenya, which are hot and they're also alkaline. So additionally problematic in DNA preservation with very different lake chemistries. However, I was able to get different organisms from these lakes, for one in water and surface sediments, and also in lake sediment cores that went back about 200 years, which is here from rotifers of the genus Brachiones, and I found 2 different species there, and I found a stratified succession of mitochondrial DNA haplotypes. So this was also intraspecific variation in 2 different sites, and for the one site we also have a story what actually happened there. So at the bottom here, we had a volcanic ash deposition, and after that, a very sudden turnover of haplotypes. So in the next sample, we had just a bit of a mix of clones. And then later, it continued. So we have another turnover later going back to this haplotype A, this also based on very, very short fragments. And in here, we had apparently more unstable conditions, perhaps at least a declining lake level, and also this other species that came in. So this was a completely different ecological situation. Nonetheless, there as well, we saw a displacement of populations. And I won't go on, but so, and I also tested for diatoms. That was interestingly much more problematic. So I did find diatoms exactly also in this lake sediment, where this was based on something called HPLC or DHPLC. And I want to say I saw that Ralph Tiedemann, who's my PhD supervisor, is also in the room. So hello, I did this there in Potsdam. And I found here, this also not yet based on next generation sequencing, diatoms only in the last 90 years. And below that, I found other taxa, which was also quite surprising. But it mirrors things that are similar to, well, kind of similar to an ongoing study that is where I generated some preliminary data some years ago, that I will briefly also show you, which is from Ethiopia, the Lake Garacha, and I got some data on from a new core that was collected in 2017 for the last 4,500 years. And as you see here, I there looked at plants, and this is all metabarcoding data now, because that was, of course, for the initial screening, the easiest to collect, and also for diatoms, also metabarcoding. And here it turned out that the diatoms, again, were harder to get, actually, even though they're aquatic organisms, than the vascular plants. So vascular plants there worked fine until 4,500 years before present. And this is similar to something that a colleague of mine, Sanne Busenkohl, I was also involved in the study, which is why I'm showing it. So this is another study where we got ancient DNA from 2 lakes in the Virunga Mountains, 2 crater lakes. And this also went back to around 5,000 years. And so this is definitely beyond a time scale of anthropogenic impact, substantial anthropogenic impact at least, and it definitely also gave some additional information. So in this paper, the stress was really on using this to provide pre-restoration targets and to look into this sedimentary ancient DNA to see what then modern anthropogenic impacts would be like. So this, in this case, it worked, but now finally, a case where this didn't work at all, and where really this was also metabarcoding data from Lake Barombi-Mbo in Cameroon, where interestingly, as you see here, the first sample that we analyzed, which was recent, gave a really high diversity of plants. But then, and this was our stupidity at the time, or we were interested in something else. We were not so interested in the last few hundred years. So the next sample started at 1,000 years, and they're absolutely 0, 0, 0, 0. There was also, luckily, hardly any contamination, but there was nothing in there. There was no more, at least not amplifiable and usable for metabarcoding. So it is very well possible that there's quite a lot in there, but it is shorter. So, however, this definitely shows that there can be a high diversity of plants in such lake sediments from Africa, which I think we should try to recover. And that is the end. And this is something I just want to stress at the end, that it's really a question, one, of time scales of analysis that vary between the different climate zones. So, and from this, I think that we have to always think about, so timescales of analysis and types of analysis, obviously, but even with quite simple methods, what we can look at in areas with not so good preservation conditions, as we will have in a lot of humid and hot environments, is exactly the last few hundred years. And so from looking at these long, really long-term studies, we can have the natural baselines, and those are really interesting and important, and they're of scientific importance to understand exactly how ecosystems, how populations work, and so on. However, things that are of immediate societal importance that have probably happened in the last hundreds of years, this kind of data can be recovered, I think, from many places without always using very expensive techniques. And that's it. Thank you.
Thank you very much, Laura. It should be very nice to go back to your past work and do an overview. Maybe you did that before, but I'm sure it's a nice feeling to say, oh, I did that many lakes in this place. So yes, as Cecilia writes in the chat, you can ask your question in the pad or directly here if you want. Again, all my questions are, you can also ask the same question than before. We can re-explain, don't worry. Took me 6 years to understand it, so don't hesitate to ask everything you have in mind. You can actually raise your hand virtually with the reaction button in the Zoom. So, Ralph, please.
Yeah, thanks for the nice talk, Laura. I like your plea for the shorter timescales, but I have a question on that, or perhaps comes a bit from own experience, but perhaps others may also comment on that. The question is, if you're in a tropical environment, what is really doing most of the harm, right? Is it immediately when the material is deposited, or is it through the aging in the sediment? I'm not sure of that. So if that were true, that it's the immediate deposit, that may then be not as easy, even in a very dry area, even to look at the shorter timescales. But if it is the aging, then of course you have a good point there to make. But for me, it's not what actually damages the DNA most.
So I think it's the aging, because so the uppermost sample from Lake Barumbi was actually not the uppermost. It was what it was like centimeters 2 to 3. However, but it was already the sedimented. It was not the fluffy layer. Okay, so this was already sedimented stuff, and that had a very high diversity. So I think that the actual sedimentation was not the actual incorporation, and the sediments was not the problem. And there, yeah, it was really a preliminary look at the data, and of course, yeah. Then Yannick and I both left Potsdam, so we have
Okay, so we have many questions in the pad. So I will start with the first one. If you are in a tropical environment,
no, that's me who wrote the question from Ralph to have a trace of it, so you can pass.
Is there a way to preserve the sediment core after obtaining them? And the question after is, where do you store your core? Where did you store your core when you took them in Africa?
Me?
Yeah.
We tried, so short cores we sampled in the field actually. And at the time we actually stored in DNA buffer and Queen's tissue buffer, which is something, so I did some tests. I would not really do that again. I don't know, Ralph, if you guys have done that a lot with sediments anymore, but yes, so we usually sample as soon as possible and then freeze.
Okay.
However, even if just kept cool, or even not cool, actually it apparently keeps relatively well. And we have had cores, of course, that have had to stay, for example, in hot summer months in Siberia for like half a year or so, and really like, and then they came and were cooled after that, and they were also still easily accessible to DNA analysis. But freezing is good, so sampling quickly and then freezing, yeah.
Okay, so to keep in time, I think we will have to continue. You can continue asking your question, and Laura, if you can answer some in the pad, that would be great. So we will continue with Abraham Dabengwa. Very sorry for the pronunciation if it's not good, Abraham. And it would be even harder with the name of the university. So from University Witwatersrand in South Africa.
It's Witwatersrand.
Great. So if you can share your screen.
Okay. let's do this. Yeah. Can you see my screen?
Yes, but you have to click on the button.
Yes.
Yes, perfect.
Which screen do you see?
Oh, you are in presentation mode. Can you switch your screens?
Wait. I could do that. How do I do? Let me.
No rush.
Let me first.
Yeah.
You can. I think you have to stop sharing and sharing again on the screen you would like to share, maybe. No?
Okay, I'm having. So okay. Okay, I'm not sharing at the moment, right?
We see the PowerPoint.
We see your screen. Yeah.
Oh.
If you go on presentation mode, then you can, I think there is an icon to just switch. Do you have two screens right now in front of you?
Yes, I've got two screens. I might as well
Yeah.
just unplug the other one.
Maybe you
How is it now?
You can just click on an icon that says switch screens once you're on presentation mode. And then normally that should do it, I think.
Okay. I think sorry for this.
It's not a problem at all.
I now have one screen, but okay. Can you see my screen now?
Oh, here we go.
Yeah, that's great.
It works perfectly.
Oh, cool. Hi. My name is Abraham. Yeah, so I want to share a bit of what we've been doing, what the research group that I've been has been doing. I don't have that much data to give, but I just want to really give an overview of like where we are and where we want to go. So briefly, what I'm interested in as a paleoecologist is environmental change, and the kind of changes that I'm interested in is the ones that are driven by herbivores, driven by climate, driven by fire, and driven by people. Why is this important? So when you look at Africa, you notice that there's a little bit of green, mostly in the middle, but most of Africa, there's a lot of brown patches. And the question is, you know, like what determines these kind of patterns in landscapes? And at the same time, because Africa has been used by people for a very long time, so these landscapes are very ancient and they've actually been stable much longer. And the question is like, how do we get to the point of talking about things like ecological restoration? You know, like, can we restore these ecosystems? Can we protect the biodiversity? Why do we need to do that? Because there are a couple of challenges that we have at the moment, like we've got climate change, we've got water scarcity, biodiversity loss, and people need to make ends meet, and most of the people actually depend on resources. And recently, because of programs to try and address climate change, there's been also this indiscriminate planting of trees in ecosystems where we shouldn't actually be having trees. So when we look at the big picture, we notice that there are places in the world which are mostly in white here. So these areas are actually those ecosystems where climate is not a very good predictor of what you find, what the vegetation is doing. And this has been bothering ecologists like in Africa for a long time, because if you look at Africa, most of it is white. And that says something about that we've got actually a complex ecology. And what some groups have been doing now is we're trying to see like how we can look at these ecologies functionally. So one of the things that you can do in the vegetation, you can see that you can pick up in terms of function, like different units. Like for instance, here you've got the mimosoid, the Combretacea, and the Deuteroides lineages. The Deuteroides lineages is one of the biggest one, the one that's like in orange. And this is where a lot of woody cover and also a lot of fire and a lot of people live. So most people are actually worried that these are actually ecosystems that used to be forests. But there's also it's an unanswered question, like where paleo actually can fit in. Are these areas actually forest or not? And then there's the issue that actually Africa can also be viewed in terms of where the herbivores are. So you can see areas whereby, they've been defined of where you can find the most animals and the ones that you can find high, like animals that are adapted to wet conditions, the ones that are adapted to dry conditions, forests, but also those ones that like those ecosystems, like that are very uncertain, where there's lots of grass. And at the same time, you can look at the continent in terms of fire. And there are many different ways that you can do this. So one of the things, like, how does paleoecological work fit in? Because there are all these theories, but we don't have enough sites to look at them. So this is one of the work that we're planning to do. So if you look, we've got a lot of. So one of the things that I'm looking at is fire, because it's one of the things that shaped this map in Southern Africa. But one of the things that you see, these are the proxies that we have. And ideally, we want to have like fire, herbivory, and vegetation together so that we can make some good like holistic analysis. But most of the yellow dots, there are very few. You can see most of it is just fire and vegetation. So we would like to at least, get this work going. So at the moment, we're still looking for funding to do the work, and also like in future, we'd like to collaborate with people who are working on ancient DNA. So where do we see DNA fitting in? So one of the things that Africa is most of the landscape has not been occupied for some time because of city flies. So these little bugs here, they were called the protectors of African biodiversity. So before DDT was introduced, there were areas in Africa where people could not live because they could actually die. And so we think that these areas where these pests were found, basically people could not go there, and those areas could actually, like, having these type of patches in the landscape could actually maintain biodiversity. So we actually don't know. Maybe we need to find the DNA of these things so that we can make a comprehensive story. There's also the differences in the functional diversity of the herbivores. So at the moment we use dung spores, but they don't give a good picture. So we think that barcoding could do, like, metagenomics could help us with this kind of thing. Same thing with grasses. Like, we'd like to see the differentiation of grasses, because there's some grasses that respond to fire, there's some grasses that respond to herbivores. We want to be able to see which ones can we, from the codes that we have, or we can work together to actually find these questions, answer these questions. And lastly, we'd like to think about how to constrain anthropogenic signals, like, crops or some fragments of human DNA that's things linked to humans. Because we can just say, oh, the fire, there was like big cereal grains and this thing, but these are just guesses. We think environmental ancient DNA could actually help us to answer good questions. Thank you very much.
Great, thank you a lot, Abraham. We will take questions even if we are a bit out of schedule, but if you have some questions, you can ask. I will try, if you can just unshare your screen.
Okay. How do I go to unstop? Like, I don't seem to see the
Stop share, I think this is
oh, I found it. Yes.
But yeah, very interesting, and of course we will discuss more about the research and how sedaDNA could be useful for your research. I think there are a lot of potential if we make it work, so that's very great. I see something writing in the chat, but
maybe we can just have one question. Yeah.
The question for you, Abraham, is can you say a bit more about the type of data you are collecting from sediment, and keep it brief?
Okay, so we're collecting charcoal, down fungal spores, phytoliths, and pollen. So those are the things that we're collecting. But we also do like isotopes, but I mean, and yeah, but that's, yeah.
That was very brief and super concise, so congrats. Maybe you can answer a bit more in the time if you want to this question. It would be super interesting for me to know exactly what kind of proxy you are using.
Okay.
And we will continue. We'll try to keep track. So for the next talk and the last one before the very short break, we will have 3 speakers, so it will be a challenge. I don't know if the 3 of you will speak or not, but we will have Rachel Meyer and Rene Larison from University of California in Santa Cruz, and Beth Shapiro from University of Rwanda, if I'm correct. So Rachel, I will let you share your screen. Yes. And yes, perfect.
Okay, good morning. I'm here with other colleagues, so I'm going to actually pass this over to Ren to kick off. So Ren, just tell me when you'd like me to change slides. And I see Tom is also here, Antoine is here. So we're a big team that just got some National Geographic funding to start doing eDNA and hopefully ancient eDNA work in Akagera National Park in Rwanda. Ren, are you there?
Hi. What did you ask?
I'm here.
Cool.
Yeah, start my video. Let's see, get on the game here. Okay, yes, I'm here fully. And I just thought it would be nice just to start, since there are a handful of us here, just if people could quickly, speak up and say who they are. I know that Drew and Beth are not here, I don't think, but I believe Antoine is here. Antoine, could you just briefly introduce yourself and say, who you are and where you're from?
Okay, yes, thank you. I am around. My name is Antoine Saimana. I'm coming from the University of Rwanda. I'm associate professor at College of Science and Technology. So I'm happy to work with the team led by Ren. And I hope it will be the first time to have eDNA in our country. Thank you.
Great, thanks, Antoine. And I think Rachel, you guys probably know Rachel, but I see Rachel did a good job of putting everybody up here. So that will kind of speed things up a bit. I believe Maishi's here. And I believe Tom is here. So if you guys want to quickly introduce yourself, or we can just jump into the presentation.
Probably should just jump in.
We'll just jump in.
Okay.
Go back to the front. Yeah.
Okay, let me go backwards. Hang on.
Okay. Great. Okay. So this is our project is called Environmental DNA to Guide the Rewilding of Akagera National Park. And this project sort of came to be when Tom and I were in Akagera a couple of years, Gruner, the manager, and he was just talking about how sort of depauperate the park was in terms of mammalian species. So there are a lot of species that, if you just go across the river 100 kilometers away, there would be, giraffes, and there would be black rhino, and there would be a variety of slide, Rachel. And so, as I noted, there's a lack of mammalian diversity, and there could be a couple of reasons for that. It could be ecological, because Akagera is kind of interesting because it's sort of been referred to as an island within an island, because you have it used to be contiguous with the Serengeti savanna ecosystem. And then I think it's thousands of years ago, maybe longer ago than that, the areas surrounding Akagera became more mesic as part of this interlacustrine environment in East Africa, associated with all the Great Lakes. But Akagera itself and the areas around it sort of retained this drier savanna characteristic. And so it's possible that animals over time went extinct naturally, but also this area has been subject to a variety of historic and prehistoric episodes of human occupation. And so a lot of these even early on, even thousands of years ago could have caused extinctions, or more recently, there have been civil wars and the genocide against the Tutsi, which have impacted that area as well. And so animals could have gone extinct locally at human hands as well. So that's what we sort of want to find out. So you could change the screen. And so we sort of want to know not only what species were present before human domination or human caused environmental change, but also what ecological processes might have been important in shaping those communities. So as we've heard, you can find out a lot about microbes and plants and things like that, and vertebrates are a little bit harder. So at minimum, we hope to find out something about the communities, but we hope to also find out about the history of the presence of, in particular, these target species, the black rhino, giraffe, cheetah, Thompson's gazelle, wildcat, African civet, the zorilla that's pictured here, the East African pato, African striped weasel, and spot-necked otter. So those are sort of the 10 species that they're really the most interested in the park, that they feel like should be there, but aren't there. A few of them, like black rhino and giraffe, have been reintroduced as of some years ago. And I think there was a presence of, I think it was giraffe kind of off and on. I think it was reintroduced, maybe disappeared, reintroduced. Anyways, these animals that we know something about the history could be really interesting to kind of verify some of our results. So since we know something's there, can we find it? Or it was there, that we can find it. And then there's a few other species of interest like hartebeest and East African oryx, which is one species of hartebeest present, but there are other species that it's thought could be there as well. And I think that's the next slide. And maybe that may go over to, that goes to Rachel.
Cool. I'm just checking that I'm not muted anymore. Okay. Thanks, Ren. Yeah. So there's a lot of lakes in Akagera, and I've never been there before, but I'm really excited to be able to core these lakes. We're going to try to get some gravity cores from 3 lakes and extract 30 samples from them. So we have a small amount of funding for this, but this could potentially lead to growing a sedimentary environmental DNA program for Akagera and for other African parks. So we're going to be using targeted PCR focused on those mammalian species, plus metabarcoding of short regions of 18S, 12S, and CO1. And we'll, you don't know what's going to work exactly until you know how long the DNA fragments are that we're going to be able to extract. And we have a workshop that is planned for December, tentatively for December of this year. So we want to get the word out. This will be hosted at the University of Rwanda with Antoine and Beth Kaplan, and we'll be recruiting students and early career researchers and park managers to join us and go to Akagera to sample sediment, then go back to the University of Rwanda and do DNA extraction. So we'll be learning all of these different processes. It'll be a couple of weeks. And then we'll process the ancient DNA. We're planning on that tentatively to be in California, but we still are really trying to make sure that we're Nagoya Protocol compliant. And as we're just starting, we recently got word, we actually submitted this a really long time ago, funded, and that's pandemic related. So we want to make sure that we have all the permits and permissions and benefit sharing plans in place, and also know how to regulate the samples and the DNA. So we're excited to kind of get into the science policy side of things as well. Just a little bit on our prior work. So I have a sedimentary DNA project in Southern California with Beth Shapiro and lots of folks from UCLA. And so we're seeing that works pretty well, except a lot of human contamination because people sometimes without gloves on. Bates kept 6 different lists, sort of capture the whole tree life. Tom Smith, who's here, has been working on that on the Congo Basin as part of the Congo Basin Institute. We have a citizen science program in California called CaliDNA, where we've been doing this. We had a small project focused on Angola short core DNA, and the samples, the way that we stored the samples after the samples were taken was not good enough to be able to ready. But yeah, and I also trying to do some protocol education. So if you're interested in that whole science policy and where eDNA falls, contact me. And I'm just going to highlight, we have found giraffe before, but never in Africa. I actually accidentally found it in California. This is the Russian River. We collected 172 eDNA samples, and just about a couple kilometers downstream of what I had no idea there was a safari in California. We found a ton of giraffe DNA, and this has been kind of haunting me, so I had to share it with you all. So we're particularly interested in what methods have worked in similar lakes. How can we strike a good balance between the optimal DNA handling and training? And we're also looking for additional funding opportunities to tack onto this to bring Rwandan researchers and teachers and students potentially to California.
That's great. Thank you very much. And I'm really happy that researchers put the failure also in the type of play they tried. Because it may be the case in the project we could launch with this initiative. It may fail also for stuff we don't handle well yet. So yes, I guess we are out of time. So maybe we can take, maybe we can keep the question. There is one question about which lake should target in the park. Maybe you can answer in the pad, Rachel. And in the same time, we can take 5-minute break. So thanks a lot, Rene and Rachel, for the presentation, and I'm sure we will discuss more about what you will do. So 5. You can come back. Okay. Researcher from the society to train a researcher from Africa. So maybe we can discuss about that, Rachel, in parallel to the one you will do, because you will do your one, and we don't know exactly how we will do the one we would like to do. But if we can merge, or if we can try to make it smooth, we should. So it's something we will discuss a bit more later.
That sounds great, Eric. Yeah, there's a lot to unpack. even where to store the cores, where to store physical samples, and DNA and frozen DNA, so that it's preserved for a long time, and how to manage that as a research resource will be really exciting to talk about.
Yeah. What will be a very good output for what we are trying to do. I mean, the lab app that we could use to do research. But it requires more than money. It requires training and a lot of stuff that is, as you said, permit and stuff are hard to get, I guess, also between countries. But yes, the questions are super interesting, and it makes you rethink everything, of course, all the time.
Eric, you need a break too. Go get a cup of coffee for a minute.
I took 3 already, so I can I can take more for today. This is quite relatively late for me. I'm actually traveling at 6 a.m. tomorrow morning to France. So I am in a packing rush time. But yeah, it will be okay. And it should be super early for you, Rachel, no? 7 or 8?
It's 8. This is normal awake time.
Yeah. Good.
Yeah, the cons, oh my gosh. It's like, as soon as you collect a sample, I'll just blab away right now. As soon as you collect a sample, there's bacteria that are eating that DNA or other, we don't know everything that's happening when you collect a sample, but if things are metabolically active, your DNA could get continuously degraded. And so keeping them cold or, you know, in a refrigerator or frozen that's why it's so preferable. So the people that were collecting the samples in Angola were in the middle of fieldwork. So they collected cores and then continued to trek on for weeks and then sent the samples through room temperature ground shipping to us, which also took more weeks. And so 2 months at room temperature, there was nothing but algae left in the samples. So we got some, you know, nothing that we were actually very interested in, unfortunately. I feel really bad because, you know, people were working really hard to get
in the fridge, and my 18S metal barcoding was just full of fungi. So it happened. It's hard. So in the pad, I put a link to the review paper we just published this year about. You may have more answers than what we can say here. Maybe some answers are not true, but it's a hard question about what are the limits collected to the sedimentary DNA. And it's a whole field of study. But,
I think we can start again. All right. So I love your dog, Beth. He's so cute. I was just seeing his little face on your camera.
Took my chair, so he's getting to sit by the computer.
It's so cute. Okay, well, so if everyone has their cup of coffee ready or tea or whatever you're drinking or not, I guess, we're going to start again. And our next presenters are going to be Fofana, Cheik, Alassane Ouattara, and El Adjissa, who come from Senegal and the Ivory Coast. And so Fofana is going to be the one speaking. He's not present today, but he recorded his presentation. And Alassane and El Adjissa will be here to be answering the questions. So I'm just going to start the presentation. And I'm just going to take this time to remind everyone to turn off their microphone during the presentation. Thank you so much. Oh, I forgot to do it. So share, and that should be slideshow. Okay.
Hello, colleague present in this symposium.
Oh.
We don't hear it. I think Cecilia closed, no?
On sedimentary DNA. I congratulate and thank all colleagues who are at the origin of this great and very timely initiative for the African session. I have honor and the great pleasure to present to you our interest in sedimentary DNA and the opportunity that offered to us, but also the challenges that await us for basic research. An overview of basic research in Africa. The case of low environmental research. Many challenges are related to a lack of infrastructure or laboratory equipment found especially in many fields such as foreign pollen, sedimentary DNA, which are very important in low environmental research. These issues are particularly exacerbated in the field of paleontology, paleolimnology, and paleoenvironment. The first consequence of this situation is that lack of major projects developed by African researchers in the field of science and ecosystems that are still poorly studied, lack of control over the evolution of biodiversity, and the availability of surface water, which is closely linked to any environmental evolution, but also to human development around this environment. Nevertheless, research time are developed with the means at hand. Our current research time, we use data by like by indicating tools due to their very varied environmental preferences and tolerance. They provide a wide range of ecology information and past and present lake environment and are therefore excellent tools for monitoring environmental change. With data like by indication tools, we uh, we create and present environment and at in science environment. In present environmental, we study of the diatom flora of Senegal, which was still limited for important hydrography system. The assessment of environmental change in space and time in the river. At ancient environmental, we reconstitute of the evolution and ancient environment around this aquatic ecosystem, which are now highly impacted by climate and environmental change. We analyze of the cause and consequence of environmental change in both and identify environment of services the final policy evolution in Africa. At sub-Saharan area, we sometimes create important hydrographic networks but very impacted today by climate change, transboundary basins that are currently experiencing both scarcity of surface water, which unprecedented environmental consequences on biodiversity, silvopastoral activity, and therefore on human development. In West Africa has fairly large hydrographic network with many rivers, lakes, marginal lakes such as delta, estuary, and lagoon. Some of these river and marginal lakes are nowadays highly impacted by climate change. There is marginal environment prevalent size for encoding climate variation and therefore environmental evolution. In Senegal, there is many examples of light past and present wetland in Saint-Like, Saint-Louis, the area, Niayes, and the Senegal River. There is many opportunity for the empowerment of African research, like set international collaborative project to boost basic research, accepting and training of research and technicians on sedimentary DNA in specialized center, tapped paleo-environmental research laboratory and strengthened research capacity in the field of sediment DNA. Reduce the dependence of African research on Western laboratory and the sometimes difficult travel to the West. Pooling of the research results on sedimentary DNA among African teams in this region. There is to strengthen the research agenda, combine the use of sediment DNA, which has of such title to achieve better results. Contribution of the science of current ecological state African ecosystems that are still poorly now. Study of other biological reconstruction with more information on science like environment. The case of certain coasts in Senegal like Tan, Redwa, Meyer area also correlate trace in the different ecosystem of the sub-region or with other distant environment and find the root cause. Research in Africa also place Africa at the heart of environment is monitoring the evolutionary trend of subtropical environment in Africa to identify the area most important. Research in Africa will therefore be a new research approach and a great opportunity to achieve results that will certainly be of interest to the entire scientific community and to African research in particular. Thank you for your attention and good symposium for everyone. Bye.
Okay, that was a great presentation. So I'm not sure if people have live questions to ask or have asked questions. Or have questions written on the pad, apparently not. Is there any questions that someone would like to ask? We can't hear
My
you.
feeling
You're on mute.
It's subjective and it's not capable of doing that. That I'm not sure about Senegal, but I have heard that from a few countries, and I wonder whether they could comment on that issue.
Yes.
Yes, I will try. Okay, I will try to answer. Okay, I'm not from Senegal. I'm from Côte d'Ivoire, Ivory Coast. But I work closely with my young friend Fofana, who gave the talk a few minutes ago. You are right. Sometimes you have some knowledge, but the problem is the equipment. For example, if you take the DNA processing, it's very difficult right now in our country to do DNA processing without problem. First, for example, how to have the DNA kits. It's something very difficult. You have to order it from Africa. The second is how to process. You don't have all the equipment in our lab to do so. So you need most of the time to send abroad our kits, our sample to be analyzed. This is something very difficult, and we don't have, let's say, funding. Most of the time, it don't prioritize, let's say, fundamental research in our country. Most of the time, it's agriculture or, let's say, healthcare or something like that. But for fundamental research, most of the time you don't have any funding. So it's very difficult in Africa to do some research like a new approach you are talking about, for example, DNA. So I think that you are in the way of construction, how to do together, let's say, to set up, let's say, a link so that you can easily go, let's say, abroad to do our research. And in South collaboration, for example, with Dakar, for example, right now I'm trying to set up extraction lab in my country, in Ivory Coast, so that we can closely work together with Senegal, because I don't have it in Fofana lab, for example. But the very big issue we are facing is equipment. It's equipment. Someone can be very well trained, but if you don't have equipment, it's very difficult.
Thank you, Alison. That's really important points and interesting. So I think Laura wants to say something.
Yes, I was looking to raise my hand in another way, but I cannot find. I don't know, my button is not working. Anyway, what I wanted to say just in reply to this latest, I mean, obviously I think the idea of the society is just this, to start collaboration, and the way you have to do is actually to start collaborate with us somehow, or with whoever you want to collaborate outside, and gradually start to search for possibility to import knowledge in your country. I can assure you that you don't need to be in Africa to have problems to start up an ancient DNA lab, because I have it myself here in Rome, like right now. So it will take years or maybe a decade in order for me to, before I retire, to get an ancient DNA lab working. But I do understand your problems, of course. So the only way to start, I would say, is to start collaborate. I know that you want to do it yourself, but the first step is this one, to start to collaborate with people. You go abroad, I'm collaborating with one of you here. And I mean, the thing we can do is just to try to exchange as much as possible. Maybe Onja, you can say something about that. You have to start somewhere, and somewhere is to collaborate. In the long run, of course, the idea is that you start wherever it's possible, a DNA lab, and start working with your own funds.
Yeah, sure. It's my case. Here in Madagascar, we have only one laboratory. I don't know, and in this lab, we have not enough equipment to do all of the analysis, or we have no DNA kits. So in this way, I contacted Laura here to help me to try to how to say that, to try to go to improve my skills first, and also to try to have results. And in this way, I think this kind of collaboration with African and with West is a very productive in French, sorry. and I think it's a better way to go sorry, I lost the vocabulary.
You're doing well, Onja. I mean, what we have done is just to start a collaboration, and with time we will find the best way to do what Onja wants to do. And I would suggest you to do the same with
those,
some other
yeah,
people.
that's so. I'm sorry to have. I will have to cut this this conversation, but I want to say that this is the reason why we actually have the second day where we are going to have those kind of conversations. So I really want to keep a hold on this, because it's super important. And that's the reason why we organize this day, these days, and this symposium with Eric, is to actually have those conversations going. So I see that there is a lot of interest, and I think they're going to be very fruitful. So because we're already late on the schedule, I'm going to have to go to the next presentation. But again, hold on to those thoughts for tomorrow, please, because those are very interesting topics. So our next presenter will be Trisha. who Trisha Spanbauer, who is going to be who is coming from the USA Toledo University. And so I'm going to let you share your screen, Trisha.
All right, I think I'm sharing, and
yes,
you should hear my audio.
Yes.
And I think it's a great transition for me to speak now, because most of my work is highly collaborative, and some of the people on this call are why I'm doing the work I'm doing, both in ancient sedimentary DNA and starting to work in Africa. I'm a new assistant professor at the University of Toledo. I am a paleontologist for most of my PhD. I am also in the middle of constructing an ancient DNA lab that you might actually hear some of that construction noise as I'm speaking. Construction is going on right now. So I'm going to be talking a bit about some past, present, and future work in Lake Tanganyika during my PhD, and the ICP had a call for scientists who are interested in working in Lake Tanganyika to participate in a working group, which is how I met Mike McGlew. Mike McGlew had been taking a bunch of short cores from Lake Tanganyika, and we did some preliminary work on those short cores. This work was also a collaborative effort with Eric Capo, one of the organizers today. He has been instrumental in really helping me develop the techniques and skills in the lab. And then I'll talk a little bit about some future work, some proposed work with some other people who are here today. I saw Ralph in the audience. And then a little bit about the ICDP program and the sort of long-term project of drilling Lake Titicaca and getting a long-term record from Africa's oldest lake. So I'll start with a little bit of the preliminary analyses that we have from the Kalia core. The Kalia core was taken from the southern portion of Lake Tanganyika. It was taken, so my work on this core was just a very rough analysis at 20 centimeter intervals of the core material. And I did 16S rRNA, a very low sequencing depth pass at this. We had very much dominance of just a handful of OTUs, but we did recover DNA, enough DNA to sequence. So I'm going to be transitioning my focus to a target capture technique on the Natiri core. The Natiri core was taken from a bit deeper waters. This core had two cores taken at the same time, dual cores. One core has been described really well, has a lot of different other proxies that are being analyzed with collaborators. And I will be doing a target capture technique on this core, the second core that's been in cold storage, in a freezer storage, as soon as my lab is finished being built. So this is like very near-term work and should be some of the inaugural work for my ancient DNA lab. Moving to thinking about the future, this is a collaborative effort between me and many colleagues who've been working in East Africa for a long period of time. And what we would like to do is take a suite of cores that really focus on different time periods in Lake Tanganyika's history. So on the right-hand diagram, this is some published data on temperature and the degree of moisture in the region. And we want to focus a food web study on different periods with paleo-environmental conditions. So focus 1 is on drought, focus 2 is on a warm wet period, focus 3 is on a warm drought period, and then focus 4 is on this high period of rapid change. So that is some proposed work that we're working through right now. And then I just want to talk very briefly about the ICDP project that's going on. ICDP is the International Continental Drilling Program. They drill all over the globe. Recently, some funding was awarded to this project from ICDP, but now the big effort is to go for these 80 plus researchers to go back to their representative countries and get more funding so that we can pool that in core several spots in like Tanganyika. At least that is the plan. What you see here is seismic profiles from 4 candidate sites. This is in the report from the last workshop. And each one of these sites is selected for slightly different reasons and to target different proxies throughout that lake basin. And I wanted to keep this brief with just a little bit of work that's sort of getting started and will be done in the future, and happy to take any questions. And I would like you to thank this group for coming together and collaborating, and also to all my generous collaborators who have gotten me to the place that I am today. So thank you very much.
Thanks, Trisha. Because we are 20 minutes behind schedule, we are probably going to have to move on to the next presentation right away, but I really want to invite everyone to ask their question on the pad, and Trisha will be able to answer as you ask. And then obviously we also have those minutes of open discussion at the end where people will be able to ask further questions. Thank you so much, Trisha. So our next presenter will be Onja Razanamaro. So she's from Madagascar, the University at Antananarivo. I'm not sure if I said it well, the name of the university, I'm sorry. Onja, do you have your co-presenter with you, Jean-Michel, or he wasn't able to make it? I think we lost Onja. Onja, are you here? No, she was here just a few minutes ago. All right, so I guess maybe she will be able to connect again soon. So what I propose is to move to the next presenter. Rene Dommain, are you able to do it, to make your presentation now? Would you be?
Yes, I can. Sure.
Okay with that? Okay. So okay, I'm sorry about that. So we're going to move on to Rene's presentation, and hopefully Onja will be able to connect after, and she will be presenting her work. So Rene Dommain is from Potsdam University, Germany, and so he will give a short presentation as well. Here we go. I let you give you the stage. Thank you, Rene.
Yeah, thank you, Cecilia. Can you see the presentation? Okay. So yeah, I'm presenting a group of scientists who work in Uganda over the past kind of like 5 years or so, and I give you some insights into kind of like the practical aspects of running such a project. And I'd like to acknowledge here in particular Morgan Andama, one of our collaborators from Uganda, and John Bosco Kubonongi, who is also in the audience today, from Barouillet University of Science and Technology. So Barouillet University of Science and Technology is our main collaborating institution. I know people there for over the past 10 years, so we had enough time to build up something. And when we got funding for this work in 2016 by the Global Genome Initiative, and in the following year, we did some initial visits to the national agencies to start the permitting process, which takes some time, and explained our research ideas to those agencies, because obviously, as you can imagine, not many people will understand what you do when you want to sample for ancient DNA, since it's a also genetic resource. So there are certain concerns. You want to make sure that you explain those things. And we did an initial field visit to see which sites we would like to sample and how accessible the site is. So eventually we came team. So research for 90 days, $50 or $100. Everyone who wants to enter Uganda needs a yellow fever vaccination. So if you travel there, you have to bring your yellow card and show this. In future, you probably have to show your COVID vaccination. I assume you can import coronavirus that we did on a refundable temporary import permit. And then you take it back and you get your money back. Otherwise, you may wait for customs clearance if you bring major equipment to the country, and you may have to pay a lot of money. Yeah. So once you're there, you can you work. So we are working in windy impenetrable forest, which is a UNESCO World Heritage Site and a national park in southwest Uganda, close to the border to Congo. And it's one of the most diverse forests in Africa and very famous for representing one of the two last remaining places where mountain gorillas occur worldwide. So it's a very important conservation site and extremely diverse. There's a lot of research going on, but there's not much known about the paleoecology and history of this site, which is the goal of our project. So because this is a conservation area, you need to get permits. And all protected areas in Uganda are managed by the Uganda Wildlife Authority. So if you want to work in such a protected area, you need first permits from the Uganda Wildlife Authority or UWA. And this application process takes at least 3 months prior to any research. You need to apply. There's an online form which you have to fill out, or an application form which you have to fill out. You submit your CV, photos, and a detailed proposal. And you have to show and you have to prove that you have local collaborators. And you need support letters from these people. Yeah, that's the first step if you want to plan working in a protected area. You have to then pay certain research fees, an application fee of $100, and then you have to put up a deposit, which you get back once you finish your project and submit your final report. Every foreign researcher who wants to work in Uganda in a protected area, for every month of visits, has to pay $250 US. Your Ugandan colleagues have to pay 100,000 Uganda shilling, which is equivalent to about $30 right now. So this is the first step. Once you have your permit, you go with this permit to the national agency which manages research projects, which is the Uganda National Council of Science and Technology. There you apply for a general research permit. For this, you first transfer $300 US to their bank account and create an online account and fill out an online application form. This will be proven by them, and then they will initiate the actual application form where you have to fill out again forms, submit a clear project proposal, photos, and so on. And this is undergoing external peer review and an ethical review, depending on your work, and also plan several months for have that being processed. When you did everything right and they like you, you get your final research permit and you can actually start working in the country. We work in a national park, so we cannot just go and start the fieldwork immediately. If you work in a protected area in Uganda, you go first to the headquarters of that protected area, in this case, the national park headquarters. And here you show again your research permit from the Uganda Wildlife Authority. And then they will know what you're doing, and then you have to get rangers and sometimes also policemen. This is for your security, yeah. So you will always have to be accompanied by these people for in these protected areas. So and as in our case, our site, our field site, was not accessible by vehicle. We also had to arrange for porters. So if this is the case for your future project, make sure you bring enough cash to pay rangers, porters, field assistants, etc. And bring a receipt book because you pay them in cash so that you get your money reimbursed back when you want to claim your field expenses. This is the practical side. Then you can set up in a field camp. We selected our study sites. This is our field area, the Mubindi swamp in the middle of the forest. We collected a number of cores. So this is field work. It took us about a week to collect all our cores. And once you're done, you cannot just take those cores out of the country. Your first thing is you go back to the national park headquarters with your samples, and they have to be inspected by the research warden. The research warden then issues you a letter, which is basically a list of your samples, the quantity, the kind of material you sampled, and so on. With that letter, you go back to the Uganda Wildlife Authority's headquarters and submit an application for research, for an export permit, which is called a research material transfer agreement. For this, you also need to show ideally an import permit for sediments or geological samples or soil samples from your home country. Yeah, so make sure you have this in place prior to do any research. And you have to pay export fees, which in the case of collecting sediment cores are expensive in Uganda, because this is not the usual material which is being collected in protected areas. Normally people collect plants or insects or blood samples. So here you see in these fees that for a kilogram of material, you pay $100 US. So you can imagine collecting 10 meters or several meters of core, you easily go into the thousands of dollars. So make sure when you submit your proposals immediately in your budget to plan accordingly ahead of time that you have enough money there to pay these things. Yeah. When you have your export permit by the Uganda Wildlife Authority, you go back to Uganda National Council of Science and Technology and ask for an export permit from them. Yeah. For this, you need to show the material transfer agreement you got, and you need to have a cover letter from your home institution that tells, or that basically says why the samples need to be exported. And we've just heard in most countries in Africa, you can say that the infrastructures are not available to of genetic analysis. So this is easily done. Yeah. So plan 2 to 3 weeks to get your export permit cleared. If you have large cores, you want to ship them by some shipping companies, or you want to take them as checked in luggage to a plane, it's really important that you also apply for an export permit from the Directorate of Geological Surveys and Mines, because these permits from UVA and from the UNCST are not so relevant for the shipping companies. They want to see that this mining directorate had inspected the material. So I didn't know that I had to run to this place just a day before my visa expired. So it was very difficult, but in the end, everything worked out. So finally, then when you have all these permits at hand, you can ship cores to your home institution or take them on a plane. What we did, because you see that it takes several weeks to get permits, we stored the cores in an air-conditioned hotel room. We were just cranked up the air condition at maximum, because obviously it's really difficult to store cores in a fridge or so. You know, these were 1 meter long cores in this case. So other things I'd like to emphasize here from a practical point of view. One thing is security. Poachers may be active in your study site, especially if you work in protected areas. This is something which we had to face. So be aware that this is an issue. Always have rangers and armed protective people with you if you go to such remote places, and also be aware that that could be serious if you bring students along. Also think that wild animals are around in most places we are interested to do our research. In our case, we had to deal with elephants. And so in lakes, you have normally hippopotamus or crocodiles. So be aware that those critters are around and may, you know, endanger your fieldwork. And then there are obviously tropical diseases. Malaria may not be a problem if you work in the mountains, but in the lowlands, it could be. So bring medication and get also trained in first aid if something happens in remote places. And always expect delays, and plan accordingly. Have long enough visas, bring enough cash so that you can survive those periods. Yeah. And my major advice is build very strong collaborations. Without collaborators, you can't do any work. And we also have to support African students, because we here in the North have all the, you know, possibilities. We have the funds and so on, which are lacking in Africa. And so, and these the students we are working with are really smart and really interested and highly motivated. So please do that. With that, I just want to point out, if you're interested in the actual science of what you're doing, there's a paper we published last year, and there's more coming out. So, and if you're interested in working in Uganda, reach out to my colleagues, John Bosco Kunurungi, who is in the audience, or Lydia Olaka from Kenya, if you want to plan to work with Kenya. Lydia was also part of this team initially. And with that, I just thank you for your attention.
Thank you, Rene. So thank you so much for this presentation that highlights a lot of important points that needs to be considered if we are starting to move towards, you know, applying like proposal application and such. So thank you so much. And again, these are going to be very important points to keep in mind while discussing the questions that we're going to talk about tomorrow during the brainstorming session. So because we don't have much time, again, we're going to move on to Onja's presentation, who just joined us back. So again, you have the pad to ask questions. If you have any questions for Rene, who can answer those questions on the pad directly. And afterwards, after Onja's presentation, we will leave also time for just discussion time for everyone to discuss. All right, so Onja, if you want to share your screen, you can, I will give you the stage. Is your internet connection fine? Is it all right? Oh, here we go. We see it.
Yeah.
It's working. Yeah. Yeah.
Oh, yeah. Like this one. It's okay?
Yes, that's perfect.
Yeah, this is on full screen. I mean, in full screen in your. Okay,
yes.
thank you. So
it's all
yeah,
good.
yeah, I'm very happy to be here for this opportunity and able to present my favorite trees here in Madagascar, the baobabs. It is not yet a subject of which we have used the sedaDNA. But so during this presentation, I will present you the ecological or biological structure of the baobabs, and a little bit on genetic study, which already been done on the baobab, because I'm a botanist, not geneticians. So So the baobabs is in the world, there are 9 species in the world and 6 are endemics into Madagascar. 2 species are in other countries in Africa and 1 species in Australia. The baobab species is belonging into a Malvaceae family and have 3 sections: Adansonia section, Longitube section, and Brevitube section. The scientific name of these big trees is Adansonia. So Adansonia section has 2 species, Adansonia digitata and Adansonia kilima. And Adansonia kilima is located around Kilimanjaro. And Adansonia digitata can be found in other countries of Africa, like Senegal, Guinea, and others. And the main characteristic of this section is that this section has a wet flower and with pendant flower and mainly pollinated by bats. For the Australian baobab, it's only 1 species in Australia, and it's Adansonia gibosa, and belonging into Longitube section. This species has a long yellow flowers and identified and pollinated by hawk moth. Hawk moth is a moth, a night moth, a night moth. So for the Malagasy baobab is belonging into 2 sections, a Longitube section and a Brevitube section. The main characteristic of this section of the Longitube section is that on this section, all of the flower of the species is very long, up to 25 centimeters in length, and have a colored flower. I already found also that on this species, all of the flower is dominated by aromatic compounds, and at this section there are 4 species: Adansonia rubrostipa here, Adansonia pereri, Adansonia za, and Adansonia madagascarensis. The area distribution of those species is mainly in the west of Madagascar, in the dry forest region. And all of those species have a large distribution except for Adansonia pereri, which species is only located in the extreme north of Madagascar. For the section of Brevitube section, this section has 2 species, Adansonia grandidieri and Adansonia suarezensis. And the main characteristic of this section is that on this section, all the species has a short flower than the Longitube section and have wet flower, but in upright location. Sorry. Those species are mainly pollinated by bats and other mammals like lemurs. The area distribution of those species also is in the dry forest in the western parts of Madagascar, and also Adansonia suarezensis is found only in the extreme north of Madagascar. So the name Suarez, it's only found in Diego Suarez here in Madagascar. The age of the baobab was already investigated by Patriot and his collaboration, and he used carbon datation during this study, and he said that the age of the baobab reached up until 500 or 1,000 years old. the main threats of the baobab here in Madagascar is the habitat degradation due to deforestation and wildfire. As we can told before, deforestation and wildfires due to poverty here in Africa. And we found also that in the scientific side that there is a low natural regeneration on the baobab. So and we found also that actually there is a decreasing on the trend of the population. So all of these threats faced the baobabs here in Madagascar. And actually, there is a high demand on seeds to produce oil from the baobabs, and the species faced on this threat is Adansonia grandideri. And it is in the scientific side a big problem, because as well as the baobabs have a low natural regeneration, the high demand on seeds will become a big threat of the species. And actually also, as you can see here in the photos, there are a disappearance one by one of some trees, and we presume that it's might be due to climate change. And actually, I have a project with Laura, and we would like to know the problem of baobab in the north, the extreme north of Madagascar, Adansonia suaresensis, because some genetic result says that within this population, they have aging population. So we make a hypothesis that there is no gene flow between this population due to habitat fragmentation. And maybe also there is a competition with another plants for the gene flow. And we would like to know this, all of this scientific data to mainly suggest to the organization, manager organization, who actually work on restoration program. So we would like to have a kind of information to give a kind of information to suggest on restoration program. As a genetic part, normally this is a part of Jean-Michel, my colleague, but as I said at the beginning, he had a big COVID last week, and I don't know what's happened right now. Maybe it's the problem he couldn't make this presentation. So I can say, but you know, he already published this data, and on this data, he would like to know where is the origin of the Adansonia digitata and the origin of other species. But he focused only on Adansonia digitata. And on this map, he said that the origin of Adansonia digitata is from the west, of Africa, and maybe transported through all the country and until Madagascar. But there is not yet a study on the origin of all of the species here in Madagascar, and the problem that he highlighted on his paper is that there is not yet a study on the paleoecological parts of the pollen, and she said that through the cpDNA, it's the result. I'm so sorry for that. And also he identified also that there is on the Malagasy species, there is a diversity, a genetic diversity, and there is a presence of a genetic introgression within species. And this is the main result of his paper. I'm not sure to know how to read this this result, but I think you can discuss it better. But I can say that at this paper, he said that within the 4 species of Madagascar, there is a mix of species, and somewhere in Madagascar, there is some population with a genetic introgression. And when he did this, study, he used a chloroplastic genomic, and he can find this kind of species, a kind of a population with introgression, with genetic introgression. So as a perspective, it's my dream, but it's not, as I said, as a botanist, I'm not sure how to be, how to say that, how to be the future collaborationist, but I'm very happy to have this symposium to tell you all about of the baobabs and all about of Madagascar. And so I'm hope my hope is to can use the sedaDNA for the baobabs or for the Malagasy biodiversity in the south of Madagascar. So thank you so much for listening to me, and I'm so sorry for my presentation for the DNA.
Thank you so much, Onja, and don't apologize. It was a great presentation. Thank you.
No, don't apologize. That was great.
So I guess we can open now to questions. So whether it's about Onja's presentation or Rene's presentation, I will give people the time
and
to.
we should, sorry, and we should start now. I mean, this is what was planned, but we can leave it open for 10, 15 minutes maybe, and after we will close it. So you can use the pad again and you can ask questions, and I will let Cecilia share. Okay. So if we have no question, we can still take some time. Maybe we can give a summary of what we will do tomorrow.
I have a kind of funny question.
Go ahead.
Yeah, like some countries declare like they have this as their heritage, thinking that a given species was, they say, is endemic from the countries. And I was like thinking from these results, from these DNA studies, we know these, the results cause conflict between countries. Like one country was considering one species like their heritage, and it's found that it was originated from another country. How do you think this kind of conflict can be managed? Thank you.
Wow.
That's a big question.
And we didn't think about that at all. But this is probably a key question. Who want to answer that? Someone working with plants, maybe, or animals?
Or it's not before me.
No, it's definitely a key question using sedaDNA, but also other tools to answer a question about legacy of species, pioneer species, and where they come from.
I mean, from what I've seen from that map, it's very difficult to say anything about the origin. That was only chloroplast DNA, and we need to know more also about, you know, gene flow and how this pollination, how does it work? I know that the baobab species, for instance, have a special time of flowering, very short, very specific pollinators and so on. So I guess it would require a bit more of genetic studies on this species in order to answer the question about origin.
Yeah. And yeah, and I guess the point of sedaDNA is not to open debate or conflict between countries.
Yeah, not to mention that I don't want to answer such questions in a kind of geopolitical scenario. It doesn't matter for me. I mean,
maybe it's a good thing we talk about that now, because if we start a project using this kind of tools, we may need a conflict of interest section about the project. Okay, about claims from different countries, and this is something we cannot handle in the society. We have no skill for that yet. So, good question, Jan.
You're welcome.
Maybe we can say a word about what will happen tomorrow, Cecilia.
Yes.
And about the form also.
Yeah. So tomorrow, how it's going to work is that we're going to have a brainstorming sessions, and we're going to split participants into 4 different rooms. One will be focusing on the ecological questions, one on founding, one will be random discussion, and one that I'm missing, Eric, do you remember? Founding, open discussion, ecological question, here we go, and feasibility, here we go. Yes. And so those 3 rooms, so you're gonna be split into those 4 virtual rooms, and you will have 15 minutes to discuss as a group. And the same as we did here with the frame pad that we shared for the webinar, you will have a frame pad for each rooms. And for each rooms, you can write up notes about the discussions, ideas and thoughts. And every 15 minutes, so you will be switching rooms, and you will be meeting new people to optimize networking opportunities. So you're not going to stay on the same room and switch topics. You will actually move to another room with other people to discuss a new topic. And so every time, so each topic, so it will be a discussion of 15 minutes, as I said. And so, yeah, the idea is really for Eric and I to be able to then, after with those frame pads, to summarize all of the discussions together, to really have a strong starting point. If African researchers wants to propose a project. And actually, Eric, if you want to say a word on how we'll next move on to and we will use this information.
Yes, I will. Thanks, Cecilia, and I will try to keep it very short. After this symposium, the first step is we will create officially the working group, African sedaDNA working group, and we will invite all the people that came today, but also people that were not able to join, to come in this group. And what we have been thinking about with Cecilia is a way to make it very efficient for African researchers to meet the sedaDNA experts they are looking for. So the way we want to do that, and of course it doesn't, it's not the case for the projects that are already ongoing, but to start new project, we will send form to people from the working group and African researchers that have an idea, ecological idea they would like to develop, like Baobab, for instance. They write a short text about what they would like to look for. We make circulate this form in the society, and the people from the society that are interested and that have the skill and lab and equipment to do that, they answer this call. If we have many people answering this call, we will send that to the African researcher, and they will decide if they want to go for an option or several options or something. And what we want to keep very, very clear and very transparent is proposals that will be written need to be written with African researcher as a PI, first investigator. This is the key here. It may be co-PI in some case, because maybe some funding will come from Europe or US or somewhere else, and you need to have someone from the country where the funding is from. But the principal investigator need to be an African researcher, otherwise we will lose completely the point of the project or the initiative. and should be first or last author of any paper that will get out of this initiative. So this is what we want to accomplish. We don't know yet where we will go, but we'll try our best. I'll be sure about that. But we'll talk more about that tomorrow. Maybe we can share a bit our idea, and you can share yours. We have many people from the society that work in Africa already and that have a lot more skills than Cecilia and I to really understand how to do that. The idea to set up. So we will use this expertise also to progress, and we'll see where we'll go. The last point is now, as this initiative, the working group, it's coordinated by Cecilia and I. And we want to change that, of course, in the future. We want this initiative to be carried out by young African researchers. So we will invite people from the working group to help us organizing stuff, and with time, if it's an initiative that is working, we will give more and more space to other researchers, and we'll just step back. That will be a very, very good change in the way we will do that. So we are still starting that. So we have to do the work with Cecilia, and we have to continue, but this is where we want to go. So please continue to use the pad if you want to stay a bit longer. Me, I will have to leave, but we will leave the Zoom open also, so you can stay as long as you want. If you want to.
And just before everyone leaves, I just wanted to say again, thank you so much for the people who participated and the speakers for taking the time to present. It was awesome.
Yeah.
So thank you.
It's better than our expectation so far. So thank you very much. It was super great. And now we hope to see a lot of you tomorrow. If we don't see you, we will share also this seminar, so you can share with your colleague as I cannot join. And we will continue discussing through the society and through the working group. So thanks a lot.
Thank you.
Have a good evening or a good day.